Back to another live stream, another video from my channel Interactive Education. And today we will be beginning with the revision session on the chapter Heredity and Evolution. Heredity and Evolution. Again, a very interesting topic, and a topic which is very important from an examination point of view. And also from studying biology, it is very interesting. So we're going to look at some of the important questions from this chapter, and we will also be trying to address some doubts. Since this is a live stream, I will be addressing some doubts. So we're going to look at the different types of questions that can come from this chapter. So I'm going to start off with that, and as people come in, we're going to start off with the doubts as well. So let us begin now with this chapter. So as per my knowledge, I have gone over many previous years papers. I've gone over many books, many different types of questions, and basically it's been seen that from heredity and evolution, the more important part is heredity, because obviously heredity, though being a small portion of this chapter, carries a lot of weightage. So we're going to start off with heredity. That's going to be my first focus. So as far as heredity is concerned, you know that heredity is what? It is defined as the transfer of characteristics. That is pretty obvious. You've understood that. It's the transfer of characteristics from parents to offsprings. Just a second. I believe that nothing can be seen here on the live stream. Oh, it's been seen that there's no view actually. There's nothing which can be seen on the thing. So let me just correct that. Really sorry for any inconvenience, because I've realized that there's nothing coming on this. Okay, Mr. Spartan, no problem. I'm really sorry for that. Now the problem is that actually nothing is coming here. So let me just fix that. So I think it should be fine now. I hope it comes now. I'm really sorry for any problems. So basically heredity is defined as the transfer of characteristics, right? That's what we see in heredity. Now we're going to try to understand what basically heredity is, and I'm going to try to address some questions which I have seen over the years, right? So one very important question which is seen from this chapter is definition of heredity, number one. Number two, they ask a lot of questions from Mendel's experiments, right? One very important question is always from Mendel's experiments. There will be one question pakka. There will be one question pakka from Mendel's experiments, no doubt, right? Now in Mendel's experiments, we have the monohybrid cross, and we have the dihybrid cross, right? They will ask you questions based on these crosses. Any one of these crosses, a question can come. Mendel's experiments, very important. Now, Mr. Spartan has a doubt in how we perform these crosses. I have shown this in my video on heredity and evolution in a lot of detail. I've explained that whole cross, right? But again, since someone has a doubt, I will be going on further and doing it again, okay? So first of all, let us try to look at monohybrid cross. Although I've done it in my previous video, I'll do it again. If you still want to have a look, it is there in the video on this series as well, okay? Monohybrid and dihybrid cross is there. But anyway, I'll do it again. Monohybrid cross. First of all, understand the definition of a monohybrid cross. Monohybrid cross basically is a cross between two organisms where we study only one contrasting characteristic. We are studying only one characteristic. When we cross them, we cross the two breeds, we are trying to study one characteristic, though they may have other characteristics which vary, but we are only studying one characteristic. So cross between two organisms with one pair of contrasting characteristics, that is your monohybrid cross, right? So that is your definition of a monohybrid cross. Now let us try to study the monohybrid cross. In monohybrid cross, what is there in the textbook specifically which is most important is for height. The monohybrid cross for height. The monohybrid cross for height we will study now. Okay, we'll try to do that. Now what did Mendel do? Mendel took two plants out of which one was tall and one was short. Now in the parent generation, this is what we are discussing, right? The first two organisms are always from parent generation. So in the parent generation, the organisms which he took were homozygous organisms. That is, their genotype had two types of genes of the same kind. So if you see capital T, this was the tall one, and he crossed this with small t, which was the short one. Right? So this is what we call the parental generation, or we call it the P generation. Okay, the parental generation or P generation. Right? So this was what he took. Okay, two plants. One was tall, other was short, and you can write this here too. This is tall phenotype and this is short phenotype. That is what can be seen in that plant. So a tall plant with a short plant in P generation. Now he crossed them. So basically next thing we are going to show is the gametes. The gametes which were formed. So what kind of gametes were formed? You will see that here you will produce gametes with only capital T, because during gamete formation what happens? Each gamete has half the number of chromosomes. If you remember, each gamete has only half the number of chromosomes. So you take capital T and this will produce gametes with small t. No other type of gametes will be formed, right? Because they have a genotype of capital T, small t. Each gamete will get either capital T in the tall one and each gamete in the short one will get only small t. Now he will cross them. So when he crossed them, these two came together. And in all the progeny, what did you get? Small t capital T or capital T small t. Right? So these two are the gametes which were produced. And this is your progeny. Or you call it F1 generation. Right? So this is what he did. Okay? Gametes and progeny, which was F1 generation. So this was the first step of his monohybrid cross. Second step of his and you see that all the plants produced were tall. All the plants produced were tall. Next, we go to the dihybrid cross. In dihybrid cross, he took any two plants from F1 generation or the progeny which was produced. So you took capital T small t and he crossed it with capital T small t. Again, these two were tall. Again, now these are the F1 generation. And he selfed the F1 generation. That is he did self-pollination in the F1 generation. When he did self-pollination in the F1 generation, he saw that each plant produced two kinds of gametes, because this time their genotypes are heterozygous. That is, their genotypes have one gene of one kind and another gene of another kind. So here we got capital T and here we got small t. Two kinds of gametes were produced. Right? Similar thing here. Capital T and then small t. So these were the gametes which were produced. Okay. Now, if you show the cross, to show the crosses, we go for a Punnett square. Now, a Punnett square is formed, right? So, if we try to draw a Punnett square, which George Punnett, sorry, used to calculate, and even Mendel used it, right? Now, this is the Punnett square. These are the gametes, right? Down and up, down and left to right and going down. These are the gametes. So here we are going to talk about plant 1, and here we're going to talk about plant 2, right? There are two plants we are crossing. So plant 1 produced what gametes? Capital T and small t. And plant 2 produced which gametes? Again, capital T and small t. Again, if you take and draw the Punnett square, this is what you get, right? Capital T small t here and capital T small t here. This denotes the gametes produced by each of the plants, right? So these are the gametes produced by each of the plants, for plant 2 here, for plant 1 here. Now capital T together will give you capital T capital T. For capital T small t, you will get here. Then here you will get again capital T small t. And here again you will get small t small t. So these were the progeny produced after the monohybrid cross, out of which 3 were tall, 1 was short. So this is their phenotype. Tallness, shortness expression which can be seen is the phenotype of the organism. So here 3 were tall, 1 was short. Hence we say that the phenotypic ratio was 3:1. Whereas if you see the genotype, there was differences. In genotype, there were capital T, there was one such plant. Then with capital T, small t, there were 2 such plants, and small t, there was 1 such plant. Therefore, the genotypic ratio was 1:2:1 when we took the ratio for capital T is to capital T small t is to small t. Right? This is the depiction of the monohybrid cross. As simple as that. Okay? So this is how we depict the monohybrid cross. If it comes in your exam, this is how you will show the monohybrid cross. Right? So I hope that's absolutely clear. For the dihybrid cross, please refer to my video on dihybrid cross. Okay, there is a separate video for dihybrid cross. In that I've explained everything, how we are supposed to do the cross also. So I really don't have time to do that now. So I'm going to go on to the things I have planned, right? And please ask any doubts if you have. Please ask your doubts. Ask doubts if you have any doubts. Okay, so please refer to my video on dihybrid cross if you still have a doubt in dihybrid cross. Okay, please refer to that video because I've explained the whole thing there. I've explained the monohybrid cross here, but the dihybrid cross, please look it up there. Okay, so I hope that clears Mr. Spartan's doubt. Okay, so now we're going to go into what I have planned. So I told you that heredity was a very, very important part. Please go over heredity very well. Please study that properly. Okay, you can get 2 questions which are very, very important in heredity. Okay, one question is, other than the crosses being asked, you can have other questions. So one question is being asked very frequently, and that question is, explain how Mendel's experiments show that traits are independently inherited. Traits are independently inherited. This is a very, very common question which is asked very, very frequently. Explain how Mendel's experiments show that traits are independently inherited. Okay. So, but if you see, Mendel performed experiments, right? In monohybrid cross, he performed some separate experiments. In dihybrid cross, he performed some separate experiments. But now if you try to study there is one thing which we see, and that is that traits, different kinds of traits are independently inherited. What this basically means is that if we have suppose the color of the seed and the shape of the seed. So for color of seed and shape of seed, they are not dependent on each other, right? Their inheritance is independent. Color of seed has its own mechanism of inheritance and the shape of the seed has its own mechanism of inheritance. So if you get a question to explain how his traits were independently inherited, you need to take the clear-cut example of the dihybrid cross, right? So how can we answer this question, right? So first point will be, answer it in points. In biology, please try to answer questions in points. It is the best way to answer. So first point will be that independent inheritance of traits is depicted by Mendel's dihybrid cross. Right? So in Mendel's dihybrid cross, independent inheritance of traits is seen. Now you know that in Mendel's dihybrid cross, we had two seeds, right? One was round yellow and one was wrinkled green. And Mendel crossed these two in the P generation, and as a result, what were all the progeny? All the progeny were round yellow, but with this kind of genotype. I'm not showing the whole cross, I'm just showing you, what all was there. So Mendel crossed a round yellow with a wrinkled green, and he got round yellow in the F1 generation. Right? So in this way, what can we understand with this genotype? What can we understand? In this, you can understand that the genotype is R small r, capital R small r, and then capital Y small y. That means it inherited one capital R gene from this, and then one small r gene from this, one capital Y gene from this, one small y gene from this. Okay. So in this way, what can you understand? You can understand that in the P generation, that is the parental generation, the genotype split. Okay, you can see that they created, during gamete formation, the alleles or the genes split. And after gamete formation, when the gametes fused, they produced this progeny, right? So they separated, right? And then they fused together to give the progeny, right? Now, this was for F1 generation. Now, when he self-pollinated the F1 generation, did he get one unanimous combination? No. If you study the cross, what did he get? He got four kinds of combinations. He got round yellow. Okay. Not like this. He got, when you cross it, and if you create the monohybrid cross, you will get the phenotypic ratio. And in phenotypic ratio, he got so many kinds of combinations. Round yellow, he got 9. Wrinkled yellow, he got 3. Round green, he got 3 again. And wrinkled green, he got 1. So this was the phenotypic ratio. You can see that he got different kinds of combinations. This is what you need to understand here. Different combinations. So since he got different kinds of combinations, here you have to understand that in F1 generation, when selfing took place, that is when self-pollination took place, that time the alleles or the gametes, during gamete formation, the genes split. Again they split, capital R, small r, capital Y, capital R, capital Y, small r, capital Y, small r, small y, capital R, small y. So you got these kinds of genes, right, in both. So in F1 generation, they split. And when they split, during fusion, different gametes fertilized each other, and that time we got different combinations. So in F1 generation, the different characteristics split and they behaved independently. Hence, we say that Mendel's traits show that traits are independently inherited. What we basically mean is that if you have a round seed, you can have either yellow color or green color, and that is seen, that is depicted here. If you have a round seed, it can be either yellow or green. If you have a wrinkled seed, it can be either yellow or green. So it's not fixed that if you have a round, you have to have yellow, or if you have green, you have to have wrinkled. Nothing like that. The shape of the seed and the color of the seed are two different traits and they are independently inherited. They have their own mechanisms of inheritance separate from each other. Right? It is a very, very important question because I've seen this come every year. How are Mendel's traits independently inherited? In this way, you have to just explain this. How they are independently inherited, you can give the example of the dihybrid cross, explain the dihybrid cross, and in the end, you have to write that different combinations were seen, not one fixed combination was seen, and hence you can say that the traits were independently inherited regardless of each other. Okay, so I hope that's absolutely clear. It's a very, very important question. If you still have a doubt, please ask me. All right, so that's one very important question which I've explained. Another important question which is asked, which is slightly smaller than this one. Okay, this question is slightly smaller than this independent inheritance, and that question is, explain how traits are dominant or recessive. How traits are dominant or recessive. Explain how traits are dominant or recessive. Again, very important question and very easy to answer. Here you need to give the example of the monohybrid cross, because it is the monohybrid cross which explains the law of dominance, right, which I have explained in my video. The monohybrid cross explains the law of dominance or the trait domination, how traits are dominant or recessive. Now in dominant traits, you know that dominant traits are those traits. How can we say that change in genes can be brought in, bring a change in DNA? Okay, just give me a second. Once I finish this question, I'll answer your question. Okay, Shitaji? Let me just finish this. So monohybrid cross will explain how traits are dominant or recessive. Now, basically in monohybrid cross, you know that we took two plants, capital T, small t, small t. One was tall, other was short. In progeny, all were seen to be capital T, small t, right? We saw that, right? After that, we saw that in capital T, small t, when we took capital T, small t and we self-pollinated them, we got four different types of plants, right? We got capital T, capital T, small t, and small t, small t. So Mendel here understood one thing. He saw that if there is one copy of the tallness trait, only one copy, even if there's just one copy of the tallness trait, the plant comes out to be tall. Even if there is just one copy of the tallness trait, the plant comes out to be tall. So in this, just explain the monohybrid cross. You can say that in both capital T and in capital T small t, there is at least one copy of tallness trait. At least one copy of tallness. Now since there is at least one copy of the tallness trait, hence the plant becomes tall. In a homozygous condition where both copies are of tallness trait, obviously the plant has to be tall. But in a genotype where only one is tall, other is of shortness, still the plant comes out to be tall. Hence it is seen that this capital T suppresses the small t. Capital T suppresses the small t and gets expressed. Hence it is seen that the tallness trait is dominant over the shortness trait. Or you can say that the gene for tallness is dominant over the gene for shortness. This is the law of dominance or the domination of traits. Right? So I hope that's absolutely clear. Again, a very straightforward question. Easy. Just explain the monohybrid cross and give this observation. Your question will be over. Right? So that's it. That's all with this question. Right? Now, Kshitij has asked me a very good question, and that question is that how can we say that change in DNA brings about a change in the genes, right? Sorry, a change in gene brings about a change in DNA. That's a very, very important question. Okay. Now, you know that beta DNA stands for deoxyribonucleic acid. Deoxyribonucleic acid, right? Deoxyribonucleic acid. And DNA is the functional nucleic acid, functional biochemical acid in life. For any human being, any organism, DNA is very important. Now, if you look into the DNA structure, okay, you're going to see that there are going to be certain strands like this. Okay. Now, I'm not going to go into the detail of the DNA structure because that's not what we're going to actually study. You need to understand one thing here. Here in the DNA structure, if you take one segment of DNA, that particular segment of DNA is what we call a gene. So gene is basically a segment of DNA. Gene is a segment of DNA. So if any alteration takes place in a gene, if any alteration is going to take place in the gene, then automatically the DNA structure will also change. Automatically the DNA will also undergo change. Because gene is a functional unit of DNA. If the gene undergoes change, that is the functional unit of the acid is undergoing a change, automatically the DNA strand, the DNA structure will also undergo a change, and any alteration in the gene will lead to an alteration in the DNA. If there is an alteration in the DNA, definitely it can lead to alteration in the characteristics of the organism. Right? So I hope Kshitij that clears your doubt. How gene and DNA are related, and any alteration in a gene will lead to an alteration in the DNA. Okay? So I hope that's absolutely clear, Kshitij. Do you still have a doubt in your mind? If you have a doubt, please let me know. So I hope that clears your doubt, because gene is the segment of DNA or a functional unit of DNA. And since it's a functional unit of DNA, definitely any alteration in the gene will lead to this. Now, what factors could lead to the rise in a new species? Okay, so factors which lead to rise in a new species. That lead to the rise of a new species. So basically, what is the formation of a new species called? What is that process called? I hope you remember that. The process of the formation of a new species is what we call speciation. The process of formation of a new species is speciation. Basically, what is this process? This process is speciation. So basically, what is being asked? We are being asked what are the factors that affect speciation? To the point question, easy and very, very important. Basic. What factors lead to the process of speciation? That is the formation of a species, formation of a new species from an existing species. So what are the factors? There are mainly 4 factors. First is natural selection. Okay, which is the phenomenon by which organisms with particular traits or with particular characteristics are selected by nature to survive in a changing environment. That is called natural selection. Second is genetic drift. Okay, genetic drift, that is the change in the frequency of a particular gene or allele which leads to the increase in the frequency of one particular gene in a population, though it does not give any survival advantage. Okay, that is called genetic drift. The third factor which affects speciation or formation of a new species is geographical isolation. Okay, geographical isolation. That is, when geographical barriers are created, they separate groups of organisms, separate populations of organisms, and ultimately this prevents them from interbreeding, and they are forced to breed within their local populations, and this will lead to accumulation of variation over time, and it will make them very different from each other. Okay, and the fourth one is reproductive isolation. Okay, reproductive isolation, which basically means that organisms will act in such a way that reproduction between them will not be possible. So if, for example, a female beetle enters a population of a different kind of male beetles, then her behavior will be in such a way that reproduction will not occur between the two. Okay, she will behave in such a way that reproduction or mating will not occur between the two populations, and hence there is reproductive isolation. So this is the four factors. These are the four factors which affect speciation or the formation of a new species from an existing species. So that's absolutely clear, Kshitij? Any other doubt you have? Any other doubt? Right? So this was again a portion from evolution. Now since we have moved away from heredity, because heredity is pretty much done, these were two very important questions from heredity. Please go over the monohybrid and dihybrid crosses back, right? So this is with your heredity, and we have discussed another concept from evolution, which is speciation, right? Okay, so let's till then yeah, Kshitij, please ask me. Just a minute. Okay, fine. Let him ask his doubt. Till then, I'll go over what I have. Now, in evolution, once we move on from heredity, okay, before that, before we finish heredity, another important concept is sex determination. Please do not forget to revise this concept. It is a very, very important concept. Okay, it is essential and imperative that you understand sex determination. If you forget sex determination, gone, because I have seen every year a question is asked from sex determination. Okay, it is very important that you understand this chapter, this concept. Okay, now sex determination, again you have to be clear with the combinations, that is XX and XY sex chromosomes. Okay, XX will give rise to a girl, XY will give rise to a boy, right? And you have to be clear with the concept of sex chromosomes. In normal human beings there are 23 pairs of chromosomes, out of which one pair is sex chromosomes. This sex chromosome pair is also called allosome pair or allosomes. Okay, and the rest of the 22 pairs of chromosomes, they do not determine the sex of an individual, and they are called autosomes. Okay, they are called autosomes. So this allosomes and autosomes, there are 23 pairs of chromosomes in a human cell, out of which one pair is a sex chromosome and is of sex chromosomes, and the 22 pairs are autosomes, which do not determine the sex of an individual. Okay, then if you have an XX sex chromosome pair, there will be a girl. If you have an XY sex chromosome pair, it will be a boy. This chromosome pair is determined by which sperm fertilizes the egg. The egg always, that is a female gamete, always carries an X chromosome, right? All eggs carry X chromosomes, but the sperm can carry either an X chromosome or a Y chromosome, right? And 50% of the sperms have X chromosome, 50% of the sperms have Y chromosome. Hence, there is an equal chance of a boy or a girl being born, and it is dependent on the sex chromosome being carried by the sperm. Okay, if an X chromosome sperm fertilizes the egg, then there is a girl, and if a Y chromosome sperm fertilizes the egg, then there is a boy. Okay, 50-50% chance. This is called genetic determination of sex. Genetic determination of sex. That means sex is genetically determined. In some organisms, sex is determined by some environmental factors and environmental cues. For example, snail changes its sex with respect to the temperature of the environment. Okay, temperature of environment. So in human beings it is largely genetically determined, but in snail it is dependent on the temperature of the environment. Okay, so that is with your sex chromosomes, sex determination, and your heredity part with this concludes. Okay, so it's pretty simple. I hope you understood it. Okay, now here comes Kshitij's two questions. There are two more questions Kshitij has given me. One is traits that are acquired during lifetime do not result in evolution. Why? Okay, very basic question. Okay, now here again you need to know about the concept of acquired traits and inherited traits. Okay, acquired traits are those traits which an organism acquires during its lifetime. Traits that an organism obtains during its lifetime. Obtains during its lifetime, right? And these traits basically involve the intelligence, the knowledge which he has gained, etc., etc. These are acquired traits. Inherited traits, on the other hand, are traits that an organism obtains from its parents. That an organism obtains from its parents, right? So those are called inherited traits. Now acquired traits, beta, you need to understand, acquired traits are those traits which do not get coded in the DNA of the organism. Acquired traits do not get coded in the DNA of the organism. Okay, so they do not get coded in the DNA of the organism or the gene of the organism. They do not they are not caused due to any coding in the DNA. No. The inherited traits are caused due to coding in DNA. So DNA and particular genes control these traits, inherited traits. Okay. That is why inherited traits will be passed on to the next generation because they are coded in the DNA and hence will be a part of the germ cells. So they will be carried by the germ cells, that is the gametes, and will be passed on to the next generation. But acquired traits are not coded in the DNA and they are coded in not the germ cells but in a special kind of cells called somatic cells. And these somatic cells do not control reproductive traits. That is, they are non-reproductive cells. Hence, somatic cells do not pass on information to the next generation, and hence, because of this, DNA coding, because of this, traits are not passed on to the next generation. So, acquired traits are not passed on to the next generation because they are not caused due to any coding in the DNA and are not controlled by the germ cells. Since there is no passing on of this trait to the next generation, they will not lead to evolution because evolution is caused by traits which accumulate over generations. And since this is not being passed on to the next generation, well, it will not cause any evolution. Understood? Kshitij, are you clear with this? Right? So that is why acquired traits do not help in evolution. They do not cause evolution. They have no role to play in evolution. Okay, so that's absolutely clear to you. Right? Now let's come to your next question, which is mutation. What do you mean by mutation? However, mutation is not there in the NCERT course for 10th standard. I will explain it anyway. Okay, mutation is basically an alteration in the gene. Mutation is an alteration in the gene. Alteration in the gene of an organism, okay, due to exposure to nuclear rays or certain biochemical substances. So mutation basically is alteration in the gene of an organism which is caused due to exposure to nuclear rays or biochemical substances. So sometimes a certain variation or a certain alteration is created in the DNA and subsequently the gene of an organism, okay, because of which certain traits are expressed, because of which certain traits get expressed, because of which certain things happen in the phenotype, certain new things come up, certain new traits come up, new characteristics come up, and this is all due to mutation, right? Now a clear example of mutation will be the Bhopal gas tragedy. If you know about the Bhopal gas tragedy of 1984, okay, an industrial nuclear plant in Bhopal, right, a particular area in Bhopal in Madhya Pradesh, there was a gas leak, a nuclear leak in the night, and because of which that nuclear waste material and nuclear material was inhaled, and all the people nearby, living nearby that industry, were affected by that nuclear radiation, and because of which even till today their generations have developed certain mutations, their generations have developed certain traits, and that is because of the alteration which took place in their gene. And to this date, whatever generation has come even to this date in that area, they still have certain deformities, certain problems, like they have polio, some of them have become paralyzed, some of them have developed extra fingers, and their face is deformed. So there are different things which have happened, right? So that is what we call mutation. Any other kind of doubt you have? Any other kind of doubt if anyone has? Right? So Kshitij, I hope that clears your doubt. Is there anything else which you want to ask? Alright, so I'll just wait for that till then. Next, we just go on to, I was discussing evolution, right? I was talking about evolution and what all important parts we have in evolution. In evolution, you need to understand the meanings of natural selection. Okay, it is imperative that you understand natural selection. Then it is important that you understand genetic drift. Okay, it is very important that you understand genetic drift and natural selection, the meaning of these two terms. You should also understand the process of speciation. Okay, speciation is important, how species are formed from an existing species. Fourth, you should understand the process of you should understand the phenomenon of acquired and inherited traits with examples. Okay, you should know examples of acquired and inherited traits. Okay, this is an important section. Okay, thanks for clearing my doubts. No more doubts. All right, no problem. So natural selection, genetic drift, speciation, acquired, inherited traits, these are important portions. Please go over them, go over their definitions. The definition of natural selection was asked last year. For as a part of a 5-mark question, there was a question, what is natural selection? So in that case, you need to explain the meaning of natural selection. Genetic drift meaning should be known. You should know the meaning of genetic drift. You should be able to explain genetic drift. Please do not depend on just definitions which your school is giving you or which the book has. You should be able to explain these phenomena, because evolution is a chapter where you don't get direct questions. Sometimes the questions are twisted. You need to explain the phenomena. In that case, you should be able to explain these phenomena rather than just explaining, rather than just defining them. Okay. So natural selection, genetic drift, you should know. Speciation and its factors, you should know. I explained it just now for Kshitij. What is the meaning of speciation and what are the factors which affect speciation? Acquired, inherited traits. Again, I just explained acquired and inherited traits. Okay, what they are and how acquired traits are not passed on to the next generation, whereas inherited traits are passed on to the next generation. Okay, so that is one more point, right? So in evolution, these 4 parts are very important, right? Next is the concept of homologous and analogous organs. Homologous and analogous organs, again very important section. Okay, because I think this is a part of your practicals as well, so there is a high chance that you will be asked a question from this concept as a part of your practicals. Homologous or analogous organs. What are homologous organs? What are analogous organs? Okay, what is the difference between homologous and analogous organs? You should be able to define them as well. You should be able to differentiate between them, and you should have certain examples. For example, homologous organs, if we see, you can give the example of 4 limbs of man and ape. of man and ape. Okay, then you should be able to, you can say the foot of a horse, and cat's paw. Okay, another example of homologous organs, right? Now for analogous organs, you can give examples like wings of bird and insects, wings of birds and insects, right? They are analogous because they perform the same function, but their structures are different and hence their origin is also different. You can also give the example of sweet potato and normal potato. Okay. Sweet potato and normal potato both perform the same function of storage of food. Both perform the function of storage. They are storage organs. However, sweet potato is a root. Its origin is of a root, whereas a normal potato is a stem. So again, their origins are different, and hence their origins are different. Definitely, they are analogous organs. Origins are different, although function is same. Forelimbs of man and ape, their structures are same, their origin is same because they have a common ancestor, and also but however their functions are different. Ape can use its forelimbs for walking, but man does not use his forelimbs for walking. Next is foot of horse and cat's paw. Again, their functions are different. Foot of horse uses it to run, whereas for your paw it just uses it to walk, right? And again, you have to say that the structures are similar and their origin will also be similar. However, their functions are different. Okay, so these are examples of homologous and analogous organs. Again, important portion. Another important portion is of fossils. Okay, you should understand what fossils are. Okay, now if you want to define a fossil, the definition of, yeah, homozygous and heterozygous conditions differ from each other. How? Okay, so that's part of heredity. I just explained it. Okay, but you should understand one thing, Kshitij. Homozygous condition, homo. The word homo means same. Homo means same. So homozygous condition basically means that the the genotype of the organism has two genes of the same kind. That is, it contains alleles of the same kind. For example, you have capital T, capital T. This is a homozygous condition. You have small t, small t. This is a homozygous condition. Because in the genotype, both the alleles for that particular gene are of the same kind. Right? That is why homo. Homo means same. If you look at heterozygous condition, it's just the opposite. Hetero means opposite. So in heterozygous condition, you will have that in the genotype of the organism, both the alleles are of different kind. Both the alleles are of different kinds for that particular gene. For example, capital T, small t. Right? So you can see one is capital T, that is one is for tallness, one allele is for tallness, while the other allele is for shortness. So capital T, small t, two alleles of different kinds. Okay? So that is your heterozygous condition. Right? I hope that's clear, Kshitij. Any doubt? This is your difference between homozygous and heterozygous condition. Right? Next we come to fossils. As I was continuing, let's come to fossils. You know, everyone has that wrong definition of fossils. Everyone says fossils are the dead and decaying matter. No, they are not. Fossils are the dead remains of the plants and animals that have created imprints on mud. That is what we call fossils. Even as per your NCERT textbook, that is the definition of fossils. The definition of fossils is not the dead and decaying organic matter. No. The definition of fossils is dead remains of plants and animals that have created imprints on solidified mud. Okay, these are what we call fossils, right? Hello Gupta Saab, hello Aman Gupta, how are you? So that is what we call fossil. Fossil is not the dead and decaying matter of plants and animals. Okay, it is the dead remains of plants and animals that have created imprints on solidified mud. That is what we call a fossil. Okay, hello Aman. You can also ask a doubt if you have. Okay, Aman Gupta. So this is what we call a fossil. Understood? So fossils is an important portion in this chapter. They will ask you the ways and methods of dating of fossils, or finding the age of fossils. So for finding age of fossils, there are two main methods. There are two main methods which they use to find the age of fossils. The first method is carbon dating. The second method is relative dating. Okay. So carbon dating and relative dating. In carbon dating basically refers to the comparison, or finding the ratio of isotopes of the same element between two fossils. Right. So when we find the ratio of the isotopes of the same element between two or more fossils, right, when we try to find that particular ratio of that particular same element between two or more fossils, we say that's carbon dating, and usually this element is carbon. Okay, usually that element is carbon. So finding the ratio of isotopes of the same element between two or more fossils, that is what we call carbon dating. The relative dating is with reference to the layers, right? So when we look into the layers in which the fossil is present, layers in which fossils are present are compared. So basically we try to compare the level or the layer at which the fossils are present. If the fossil is present at a higher layer that is closer to the earth's surface, then we say it is a more recent fossil. But if the fossil is present really low in the earth's crust, somewhere very very low, then we say that it is what? It is an older fossil with respect to that particular fossil which we discussed earlier, closer to the earth's surface. Okay, so carbon dating and relative dating, two very very important methods to find the fossil age. Okay, and this is asked very very frequently, that you need to find the age of the fossil, either carbon dating or relative dating. Please be clear with this. Okay, so that's with that fossils. Then evolution by stages. They usually ask you in evolution by stages two very very important parts. Okay, one thing what they ask is the evolution of eyes. Okay, in evolution of eyes you basically need to just explain the evolution of eyes and the different kinds of eyes which you find in different organisms, right? Whether it is an octopus, then the planaria, then reptiles, then different kinds of animals have different complexity of eyes, and we compare them and we try to say that just on the basis of their eyes we cannot classify them into different groups or in the same group, right? So this is the evolution of eyes, how eyes have evolved. That's the important part. Second part which you need to discuss, and second part which you need to study in evolution by stages, which is important, is the relation between birds and reptiles. So how birds have evolved from reptiles, that is an important portion, right? It is very very frequently asked. Explain how birds have evolved from reptiles. In this case you just need to talk about feathers and wings. Okay, how feathers they developed their feathers to for flight, and reptiles, some reptiles also had the ability to fly. Okay, some also had feathers. So in this way we say that that birds have evolved from reptiles, and birds have evolved from dinosaurs, and dinosaurs were ultimately reptiles, hence we say they have evolved from reptiles. So relation between birds and reptiles. Okay, so basically we try to find the relation between birds and reptiles. It's an important question, please be clear with this. Okay, that's evolution by stages. After evolution by stages, evolution of wild cabbage, which is very rarely asked. I have not seen the evolution of wild cabbage, okay, being asked very frequently. Evolution of wild cabbage, okay, which is an example of artificial selection. Okay, please look up my video on artificial selection and evolution of wild cabbage, then you will understand what I'm talking about. So evolution of wild cabbage, again, I have never seen this being asked for a very, very long time. However, please go through it. You never know. CBSE is a very unpredictable organization. Sometimes they ask you things which you have never expected to be asked. So evolution of wild cabbage, please just go through that. Artificial selection, please go through that. Okay, just a bit, just a eye wash of that. Just go through it once. Okay. So evolution of wild cabbage, artificial selection, just go through that. Then another question which is very important in this chapter is why evolution is not equal to progress. Evolution not equal to progress. Again, important question. You need to know this. Why we say that evolution and progress is not the same thing. Evolution and progress is not the same thing because organisms may have evolved. But we don't say that a particular organism is higher. No, the concept of higher or lower or more progress, that is not right. Okay, what we what we say, what we argue to prove that evolution is not equal to progress is that even if organisms have evolved, but older organisms still survive. Still survive. It's not that they are dead. No, they are not dead. They are still surviving, right? They still are surviving in this changing environment. Organisms may have evolved, but the older organisms still survive in this environment, still survive in their respective environment. That is why we say that organisms may have evolved, but older organisms still survive, right? That's organisms evolution and how evolution is not equal to progress. You can give the example of bacteria and say that how different organisms have evolved from this very, very primitive bacteria, but still bacteria has been able to survive in changing environments such as hot weather, such as change in water conditions, and still it is able to survive in small regions and such regions where other organisms cannot dream of surviving. So bacteria in one way is still existing, is still surviving in its respective habitat. So you cannot say that evolution means that the organism has progressed or has, developed. No, that's not biological development. Evolution and progress are not the same. Just keep that in mind. And these are the facts why they are not the same, because organisms have evolved, but older organisms still survive, right? Example of bacteria can be given, how it lives in small niches, that is small living spaces, and such spaces where other organisms cannot even dream of surviving. Okay, so that is why evolution is not equal to progress. Another important concept. And the last concept in this chapter is human evolution. Not too very important, but still human evolution, just go through it. Okay, just go through the concept of human evolution. Every human being is part of that same family of Homo sapiens. Okay, everyone is part of that same species of Homo sapiens. Homo sapiens is that important species. No organism, no human being in this world is not a Homo sapien. Every human being is a Homo sapien, regardless of the change in color, texture, color of the skin, the different body built. All these are different perspectives. But Homo sapiens is that one species of humans which no one can change. It is the common species, right? We still classify them on the same in the same species regardless of the differences that they have, and that is because they are part of the same species of Homo sapiens and they are able to breed amongst themselves. Right? Regardless of their differences, they are able to breed amongst themselves, okay, and perpetuate, produce fertile offsprings, right? And hence we say that human beings, though they have evolved in different regions of the world, they have walked to different areas of the world, yet they have developed different traits, different characteristics, yet they are part of that same Homo sapiens species, and hence we say that they are part of the same species, right? So with that, I have covered more or less your human evolution, and I have covered your evolution by stages. Sorry, I have covered this heredity and evolution chapter, all the things I wanted to tell you. If you still have any kind of doubt, please post it in the chat box. If you have any kind of doubt in this chapter, don't hesitate to post it in the chat box. Right, I'll be waiting here for some time. If there is any doubt, you may post it. Otherwise, I'll close this stream. Whew. I think I'll just close the stream now here. Thank you very much for joining me today. I wish you all the best for your exams. If you have any kind of doubt on this chapter, please post your doubt in the comment section below. Thank you very much for joining me. Goodbye. Stay healthy, stay smart. Best of luck for this test. Best of luck for this chapter. Easy chapter. Just go over the concepts. Bye bye. Stay healthy, stay smart, and do keep studying.